So thank you, Scott, and thank you all the young academy for giving me this opportunity to speak a little bit about infertility and the effect or the relationship that the sperm cell has. So I will try to be very succinct. So then, if you have any further questions, so I will happy to answer them. So the first thing that I would like to mention is that what we understand about fertility, for sure refers to the capability to conceive or induce pregnancy, and infertility is a term that refers when a couple that is in the conceiving years tries to give birth a child, but after trying that for 1 year without sexual protection during the intercourse, they are not successful. So when we are facing this kind of infertility, and we will refer about primary infertility, which is the one that is associated to a problem in the gametes, the oocyte in the case of the female, the sperm in the case of the embryo, we have to say that between 10 and 15 of couples are infertile in the Western world, which is a very, very high proportion if we take into consideration, or if we keep in mind the frequency of other diseases. So this is considered by the World Health Organization as a disease. And if we look into which the reasons or which the cause of this infertility are, we can first say that 20 to 30%, this depends a lot, or this relies a lot on studies, depends or is related to a male factor. A similar figure is related to female factors. 20 to 40% is due to both male and female. And then we have still a high number of cases in which we do not know why that couple are unable to give birth. Regarding the male factors, which as you can see is 20 to 30%, we have to say that, as you can imagine, they are directly related with the sperm. The sperm cell, the spermatozoon of the sperm cell, is a very particular cell, which across the mammalian division, and particularly in the Eutherian placental mammals in which we belong to, it's a cell that has a head, has a tail, okay, and most of the organelles that are used to see in a normal somatic cell are absent in the sperm cell, but yet we can find a nucleus where the nuclear material is kept, and then we have the acrosome, which is very important for the sperm to fertilize the oocyte, and the mitochondria, which are also very important to control the sperm movement and a process that is known as sperm capacitation. So the cell is very important for when that cell has to reach the oocyte. But what happens after the sperm fuse with the oocyte? So what happens after fertilization? Does that sperm cell have any further role? The response is yes, there is a role for the sperm, and we have not paid much enough attention to that. And embryologists and gynecologists have always been paying more attention to the oocyte and female factors than to the sperm ones. So there are components of the sperm, the proteins, the DNA, messenger and microRNAs, and also the centriole that do have a role after fertilization. So after fertilization, these components, we can identify the sperm components after fertilization. We can identify the sperm mitochondria, although they are quickly destroyed. We can find of course, the DNA, but we can also find sperm proteins that are very important in the activation of the oocyte. So in a process that occurs after fertilization and is driven by the sperm cell. So if the sperm cell is unable to trigger that process, we will have fertility case that apparently is not related to the sperm cell dysfunction, but it is. For example, we can identify different sperm proteins in the embryo. Okay, we have a list of sperm proteins, up to 59 sperm proteins that are present in the embryo, especially in the first stages before reaching what is known as the blastocyst stage. And I would like to draw the attention on a particular protein which is called PLC zeta, okay? Because this protein is a protein that regulates and drives the process known as oocyte activation. So this means that any oocyte that leaves the ovary and is not fertilized, so any oocyte that is eliminated through the period during the menstrual cycle, is an oocyte that has not completed the meiotic division, because it has been arrested at the middle of the second meiotic division. Only if its oocyte is fertilized and is fertilized by a sperm, this oocyte will complete that oocyte division, that second meiotic division, and this is a process driven by this protein known as PLC zeta. And this protein mobilizes and triggers a signaling pathway, very particular, very specific, that drives the oocyte to complete that division and then to start the meiotic division of the new zygote, so after fertilization. If it has been very apparent that mutations, and this is something that we have been studying for quite a long time, that mutations in this protein drift to a complete fertilization failure of the oocyte, and despite the fact that apparently the problem is not the sperm problem, because you can see how the sperm move, how the sperm definitely work in a normal way. It is able, the cell, to fertilize the oocyte, but then nothing happens regarding to the activation. Here, one could think that the problem is due to the oocyte, but it's not the case, because the problem in that case is due to the sperm. If we look into, if we compare the reasons or the factors that are related to fertilization failure when IVF, which is the conventional in vitro fertilization, and ICSI, which is the intracytoplasmatic injection. This means when we take the oocyte with a pipette and then we inject into that oocyte a sperm cell. This is the ICSI. So when we do one of each of these assisted reproductive techniques, and one of them, and each of them fails, the reasons about this failure are different. And take into consideration that when the intracytoplasmatic injection fails, so this means that we take the oocyte, we put the sperm in, and this fails, it is mainly because a sperm factor, a male factor, not a female one, and in most cases, this is related to this particular sperm PLC zeta protein. Another very important point that we should to look at is the integrity of the DNA. The DNA of all cells is organized in something that is known as chromatin, and this chromatin is the association of DNA and proteins. However, these proteins, in the case of the sperm, if the ejaculated sperm, are different from the normal somatic cell, because most of the proteins that are present in the sperm, in the normal chromatin, in the somatic cell chromatin, which are known as histones, are replaced by other proteins that are known as protamines, and the organization of the entire sperm genome in a mature sperm cell is completely different. There are, however, in this organization, which is mainly organized into what is like a donut or toroid, and between these toroids, there is a region that is called toroid linker region that is very susceptible or very sensitiveness to the environmental changes and can be broken. If this region is broken or is fragmented, we say, we have what is known as a sperm DNA fragmentation. And this is also a cause of male factor infertility. However, in not always it is apparent that this DNA fragmentation has a direct impact on the fertilizing ability. So, for example, we have different reasons that lead, or different causes that lead to sperm DNA fragmentation, but this sperm cell is able to fertilize the oocyte. So there is a oocyte fertilization. We can see in the oocyte both the male and the female pronuclei, and if there is a minor DNA damage in a sperm that was being burned by the sperm and is inside the oocyte will try to repair that DNA damage in the sperm. So the oocyte will take its own machinery to repair the sperm DNA damage. If this damage is massive and the oocyte is unable to repair, this will enter apoptosis and everything will stop. This means that the oocyte will stop the process and we will have no embryo implantation because the process will have been stopped. However, if there is a mutation, a very big mutation, here we have a problem. And this problem can be seen in this clinical data. Here we have a diagram that we have the DNA fragmentation. We have a comparison between the in vitro fertilization and ICSI, and looked at the abortion rate that is observed when each of these techniques is employed, and when it's a very big sperm DNA fragmentation, and we conduct these techniques, because this technique ICSI bypasses a very big amount of barriers, that abortion rate is higher. So this means that if we have a very big sperm DNA fragmentation, this DNA fragmentation perhaps cannot lead to a total fertilization failure, but can ultimately lead to abortion. So this means that some miscarriage that occurs in couples that have been submitted to assisted reproductive technique protocol are not due to the female, but the male factor. So that problem appears later. This is also very apparent in this other data, in which you can see the implantation rate. So the low end implantation rate, low DNA fragmentation has a higher implantation rate than the high DNA fragmentation has. And when we compare the female age, we can also see how, if the female is older, more than 30 years old, the number of events of implantation are also lower, which indicates that here we have a couple of problems. We have a sperm that has a very high DNA fragmentation, but at the same time, we have an oocyte that is more aged, and because of that, that oocyte is unable to repair that DNA damage. Finally, I also wanted to speak about the RNAs and the microRNAs, because we have RNAs, which is a material that theoretically should not play any function in a sperm because that cell has no transcriptional activity, yet we can find a specific sperm transcripts in the after fertilization. So this means that a sperm not only delivers the DNA, but also the mRNAs, and these mRNAs definitely decrease over the first embryo stages, but they do play a role. This means that if we have dysfunction in those mRNAs, we can also have some cases of infertility, and again demonstrates how important or how much important a sperm is for fertilization events. And finally, regarding the microRNAs, it has been reported that exposure of the male to an environment that is a stressful environment, or toxic factors, an altered diet, smoking, and so on, can affect the microRNA cargo that the sperm have, and this can have an impact on the epigenetic changes or epigenetic marks that this sperm cell has, and this also has an impact on the health and on the fertilizing ability of the offspring. And this particularly regards to microRNA, so small molecules that regulate the gene expression after oocyte fertilization, that have been identified over sperm maturation and production in the male, and then they have also been identified in the oocyte. So in conclusion, just to say that the evaluation of the sperm cell over the next, the previous 30 years has evolved a lot. And despite the fact that initially we paid more attention on whether or not the sperm were viable or were motile, and we're thinking about only this particular spec, we have also then seen that this is much more, the contribution of sperm is much more than the DNA and the fertilization. There are some components of the cell that have an impact beyond that, and this means that some infertility-related disorders, although they can initially be attributed to the female, are directly related to the male. And with this, I am done, and I hope that I have not consumed too much time, Scott, and thank you for your attention.
Thank you very much, Marc. Fantastic, another fantastic talk on a very different topic again. So we have a little bit over time now, of course, but these tend to go over time by a few 5 to 10 minutes. Gemma, you have a question.
Yes, thank you, Marc. That was such an engaging and clear presentation. Thank you. Just a quick question about, I mean, this is really not my field, but so you said that if there's sperm damage after fertilization, the oocyte will deploy some sort of mechanism to try and fix that. I thought that was very sweet and very, very heartwarming. Do you know how these mechanisms look like, so that you can reproduce them artificially to try to rescue the damage?
Yeah, we have some idea about this DNA damage. Basically, if we have DNA fragmentation, and this DNA fragmentation is a single strand fragmentation, this means that it just affects one of the strands of the DNA. The oocyte just takes the other one as a template to repair that. Because here the problem is that all cells, every cell, has a DNA repair mechanism. And some of the problems that can ultimately lead to cancer is an incorrect repair of this DNA. However, sperm cells, because are a very simple cell that has not many things that other cells have, the sperm cell does not have this mechanism. So it is enabled by itself to repair its own DNA because it is devoid of that mechanism. It has been removed because the idea here is just to produce a cell that is very small, and the smaller the cell is, it is better or it is easier for them to be moved, but at the same time, it has less mechanisms to repair that DNA damage. So the oocyte has the mechanism, and it takes that single, if there is a single DNA damage, it can repair. But this is limited. This is limited. So this means that in the case of the double-strand DNA fragmentation, it is not clear to which extent it is able to repair that. It seems that the younger oocytes, those of women younger than 30 years old, we don't know enough yet, or we don't know enough in detail about which are these differences, but they have more ability to do that. So this is something that needs to be investigated. And then regarding your second part of the question, how could we translate that into artificial rescue? That's something that is a little bit more complicated because the sperm cell is a very compact and packed cell. So it is very difficult to get in the cell and repair that. So at this moment, the only possibility that that I believe that can be is intervening not before but after fertilization, when we can intervene in the again against the sperm DNA damage, but within the oocyte. But the problem here is that we cannot know how does that sperm DNA look like before, because we will be killing the sperm. So that's a very difficult situation, because in most cases this high DNA fragmentation is linked to an inherent genetically disease, and this is very, very difficult to rescue. So semen donor in most of the cases is advised.
We have Christina who's raised her hand, and I think we have, so that we're not going too long over, Christina, if you can keep it a very short question, and Marc, a very short answer, please.
It's related to the first question, so I'm sure it would be a very brief question. And I was also very, very intrigued and very surprised by this self-repair mechanism. So I want to ask you if this issue of healthy habits and a healthy lifestyle helps to the self-repair possibilities, if there's some kind of interaction.
Because there are some kind of sperm DNA damage that is induced by those environmental stress factors. A sperm DNA fragmentation, when it is linked to this oxidative stress environment, can be treated by giving the male antioxidants, pills, for example, and it can also be treated, or there is now evidence that specific types of diet, like the Mediterranean one, which is rich in omega-3 polyunsaturated fatty acids, helps. So the diet and the Mediterranean diet, not smoking, not taking alcohol, helps when the DNA damage is related to the stress, not definitely when it is genetically derived.